last talk of this session of our Water and Exposure Science section, we have Carr here who's going to be speaking about the Binational Early Asthma and Microbiome study. And then you'll also see Tara later for our panel. So, whenever you're set up,
is that working?
Yeah,
okay, great. Thank you so much. It's really wonderful to be here. I'm Tara Carr. I'm an associate professor of medicine, and I'm an allergy and immunology physician, and I take care of a lot of patients with asthma, and that's where my research is. Asthma is the most common chronic disease that affects children, but it also affects a lot of adults, particularly this time of year when the trees are showering yellow toxins. So I'm delighted to share with you BEAMS, or the Binational Early Asthma and Microbiome Study, which is currently funded by the NIAID as a program project grant and is ongoing. So obviously, right, the environment influences health, and that's why we're all here. And we're particularly interested in the microbes of the environment. And as it pertains to asthma and allergy, there's this really great story and great example of how this can happen through the Amish and Hutterite comparisons of health. Amish and Hutterite populations here in the United States kind of came from a founder population in Europe. They have very similar family structures, very similar social structures, but they farm very differently. The Amish people farm with very traditional farming, lots of farm dust and microbial exposures, whereas the Hutterite population has much more mechanical industrial farming. And it turns out that all of that farm dust exposure is good for you and protects against the development of many diseases, including asthma and allergies. And our group, along with others, showed that this was actually due to the way it influences the immune system, which is very exciting. Another really important example is this the Karelia area between Finland and Russia. This border was increasingly separated after World War II, and so this population kind of got divided. And on the Finnish side of the border, there was a lot of sort of industrialization and modernization, but that went along with an increase in the rate of allergies and asthma in that population, which was not seen on the Russian side of the border, where there was less industrialization. And some photos taken here of the Russian side of the border really emphasize that there were lots of differences in the environment, including, importantly, the drinking water. And these kind of rusty, old-looking sinks are where people would drink from. And the microbes that are in that water and in the dust were linked to protection against asthma and allergy on the Russian side of the border. So we do believe that there are some microbes that are indeed good for us, and this is sort of that hygiene hypothesis. And so we want to learn more about what those are. So the sort of framework of this is that there are lots of things in the environment that a human experiences, starting as early as our first environment in utero. And you know that when you're born, you have lots of exposures. In those first few months of life, we have a lot of exposures. And we believe those exposures really set you up for what your microbes are going to look like in your body. And that can be influenced by things like antibiotics that you're exposed to, pets in the home, animals, but then the food that you eat, the water that you drink, and the dust that you're exposed to and other environmental exposures. So these things ultimately will influence the types of microbes that are living in and on our bodies, but also the way those microbes are functioning. And those things influence the health that we have through potentially the products that are being released by the microbes, and the barrier function of our guts and our lungs and other areas where the microbes are present. So we live in an area where we have a border that's had increasing border restrictions and sort of separation of populations over the past decades. And we did an assessment of asthma rate among middle school age children in Nogales, Mexico, so right across the border, and then in a few schools in South Tucson on this side of the border. And we showed that on the US side, there was almost 4 times higher risk of asthma than there was in Nogales, Mexico, which led us to say, okay, we have a border, we have a population, and I'm sorry, this was limited to children of Mexican descent. So we have this population, we have a border, we have separation, and we know that there are differences in the way that we live in the US versus on the other side of the border in Mexico, not only in the way the communities are structured, but in the way that people obtain drinking water and the source of their drinking water and the size of the homes and the number of pets in the homes, a lot of other environmental differences. When we collected samples of drinking water and house dust from homes in Mexico and homes in Tucson, we saw that there were very different microbes present in the drinking water and in the dust when you compare those two locations. And interestingly, if you if you look at the stool of a 1-month-old baby, already at 1 month of age, the stool contains microbes that are present in that house dust. And when you take the acellular products, the water product of that stool, and put it on cells in a lab, you can show that it can influence the way those cells develop towards an allergic or a non-allergic type. So basically can influence the biology. So even at 1 month of age, we're reflecting our environments, and that reflection is really influencing the way our immune systems are developing. So the BEAMS cohort is asking what environmental exposures, prenatal, perinatal, postnatal, are protective against asthma development. And, you know, what's the difference between Tucson and Mexico, and how can we sort of understand better what's happening so we can harness it towards ultimately protecting babies and children from getting asthma. So this is a birth cohort. We are enrolling pregnant women in Tucson, and we are enrolling pregnant women in Nogales. And so we really do have a binational study here. And what we're expecting to see, and our hypothesis, is that there are different environmental exposures in both locations. These exposures influence mom, and the exposures in the mom influence the baby. And then in the United States, we're going to be more likely to see sort of a restricted microbial development that's maybe a little bit less healthy and leads to more allergy and asthma. And in Mexico, maybe a more diverse, healthy microbiome that leads to health. So to do this, we are identifying and recruiting pregnant women of Mexican descent in Tucson and then in Nogales through our clinical partners, El Rio Health and the Centro de Salud. We are undertaking home visits during which we collect health questionnaires, food questionnaires, but also human samples like stool, blood, nasal samples, vaginal swabs, and environmental samples, house dust, and drinking water. And we're collecting birth samples from the baby, and then we're collecting samples from the baby in the home for the first 2 years of the baby's life. We have a couple of surrogate markers of asthma at 2 years of age, which is kind of like allergy with wheezing. And we're doing a lot of immune assessment, both from the mothers and from the babies, to try to understand how the immune system is developing in relation to the environmental exposures. But the sort of other layer to this is the microbiome, and how we believe that the microbes in the environment are influencing the baby and the mom. And Sue Lynch at UCSF is driving a lot of this work to not only look at what's present, but how they're functioning. And then Donato Vercelli is going to use mouse models, germ-free and gnotobiotic mice associated with stools of both locations to really sort of prove our concept. We've been recruiting for about 1.5 years in both locations. Moms on the U.S. side of the border are a little bit older and report a little bit more years of education than moms in Mexico. And moms in Mexico report fewer episodes of asthma diagnosis and related symptoms than the moms in the U.S. Home environments are different, as we know, and the source of drinking water is quite different. 20% of moms in the U.S. report drinking from the tap, 52% from a sealed bottle, and 1/4 hauled in a container, whereas almost all the moms report hauling their water in Mexico. And our water is collected in kind of big containers, sterile processing. Dr. Beamer in the back over there, hi Paloma, and Carolina Quijada from her lab have been spearheading this. We filter the water to collect those microbes, and then we're storing aliquots of that filtered water for future analysis. To just show you the difference between a sealed bottle, like these big bottles that get delivered to your house with the seal on it, or that you buy from the store, versus the hauled water, which are these garrafons, which are refilled regularly without sort of sterilization in the middle. And see, they're not covered, and they're refilled at these purifying machines or the purifier stores, just so that people can have water to bring to their homes. So we're really interested in what microbes are present in that drinking water, if those microbes are different by location, by source of drinking water, if we can find those microbes in the humans that they're drinking that water, and then if that microbial exposure, is it all related to the health outcomes that we're interested in, the immune development and the presence of wheezing and asthma risk in these populations. But of course, there are a lot of other questions we can ask about water, and I'm kind of excited to hear opportunity for collaboration in this group and across the University of Arizona. We'd love to know what metals are present in that drinking water, because of course metals can directly influence health, but metals can also influence microbial function. And if we understood better what metals are present in the water by location, by source of drinking water, we can get a sense of what that risk is going to look like, and then how that influences the health outcome. And then of course we can combine that with some of the microbial data to understand how the presence of metals really influences that microbial effect for health. This is a binational program. We have a binational team of researchers and scientists who are working on it. I'm delighted to be here to represent the group, but I will acknowledge that there are a lot of people who have been actively participating in this study and really move it forward, and to acknowledge our funding sources and then the many investigators who are involved in the study. So thank you so much for having me.
Thank you, Carr. We have time for questions.
Thanks so much. This was really interesting. Looking at the environment, are there going to be any surveys that include pets in the household as well?
Yeah. Thank you for the question. Absolutely. We struggled because we had so many things we wanted to learn about the environment because, of course, when you're developing questionnaires and you're developing a research program, you sort of have your a priori hypotheses, but we acknowledge there's so many things we don't know. And so we're trying to collect as much as we can, but have it also be feasible. But we are collecting number of pets. There are differences in the number of pets that are in the home, but also that are indoor-outdoor pets and outdoor pets specifically. And so we're capturing all of that. We're also capturing measurements of the home, like number of bedrooms, number of bathrooms. When we vacuum the dust, it's in a standardized way, so we'll be able to standardize to home size, type of flooring, presence of mildew on the walls. So we're getting as many environmental characteristics as we can. We have GPS locations, so we'll be able to do some identification of particulate exposures using the GPS. So yes, and we're really excited about pets. So the research center that I'm part of, which is the Asthma and Airway Disease Research Center, is a center here at the University of Arizona Health Sciences led by Fernando Martinez. And they've been doing research in asthma since before I was born. They were one of the first groups to report that dogs in the home are actually protective against asthma. And at that time, nobody knew what that meant. Microbiome wasn't really a thing, and certainly we couldn't study it nearly in the way that we're able to now with all the technology that's developed. So we are really excited about the way that those animals influence our exposures, pets and beyond, and certainly with the farming communities and some of those other the other ways that we see this diverse microbiome really influencing health. We want to capture that info.
Yeah, and asthma is one of the most common diseases in our cats as well in the upper respiratory system. And so a lot of the mechanisms that we produce out of home for the cats end up benefiting the children that have been affected as well.
I love that. That's great. As an allergist, I'm always, environmental controls are great, and if it benefits the whole family, including the furry children, that's great too. Thank you.
Just a comment stimulated by your showing the Amish Hutterite 2016 New England Journal. You're probably aware that in 2000, Tom Ball and others here in Tucson looked at the same issue in daycare centers, ones with siblings and kids with siblings and kids without. And this has been cited 1,322 times, according to what I just looked up.
Was that with the Tucson Children's Respiratory Study or with the Infant Immune Study? Oh, sorry. Was that a study in Tucson, or was that here with the research? Was that the Infant Immune Study with Dr. Wright?
No.
Oh, okay. I think that was around the time that we were also publishing from one of the cohort studies that there was absolutely an influence on daycare and sibship.
No, I think they're all in agreement.
Absolutely. Yeah, it's interesting. I think the idea is that the more exposure you have to good microbes and good diverse microbes, the better. And sometimes the places where you get lots of good microbes are also the places where you're exposed to things like viruses that can make you sick. So it's hard to tease that out, but I think you're right. It's really fun to be here where there's so much history in this.
Just another personal aside, I worked in Papua New Guinea for 6 years in the 70s, and there was no asthma.
Yeah.
In 1988, when I went back for a sabbatical, it was starting to show up in the children of the hospital staff who had cold and water noses.
That's, I think, another really great example of how the environment influences health in a negative way. Yeah, it's a great observation. Thank you.
So the last observation that's never been published is growing up in Alaska, we had a hay farm. and yet I'm a hay detecting machine. I start sneezing seemingly miles away from. So would it be worse or better if I had not grown up baling hay?
It might have been better if you had not left the hay.
Okay. Thank you, Tara.
You're welcome.